Assalamualaikum and a very good morning, everyone. We are Group 4. My name is Nur Amira Shuhada. Our topic is occipital lobe, and I'll be doing the introduction as well as treatments of damage towards the occipital lobe. Introduction. Occipital lobe is located at the posterior portion of cerebral cortex. Is this lobe mainly responsible for our vision, for human vision? It covers 12% surface area of the neocortex of the brain. It produces electrical stimulation via visual sensation. Damage towards occipital lobe would lead to complete or partial blindness, or also there is a disease or it can be diagnosed as visual agnosia. So fun fact, people who are wearing glasses like me are actually legally blind. So another two lobes that works together with occipital lobe is parietal and temporal, and these three lobes works together with our cognitive processes, will influence our visual perception.
Greetings to Dr. Farhana and fellow friends. I am Amanda Vivian. Now I will be presenting about the anatomy of occipital lobe, which consists of two parts. The first part is medial and lateral, and the second part is the subdivision. As you can see in this slide, this is the whole structure of the human brain from the top view and the side view. The part which is highlighted with red color, is the occipital lobe. So we can see that the occipital lobe is located at the furthest back part of human brain. Moving on to the medial surface and lateral surface of occipital lobe. So the medial surface of the occipital lobe is delineated by the parieto-occipital PO sulcus, and the lateral surface of the occipital lobe is delineated by parieto-temporal lateral line, which is an imaginary line connecting the end of PO sulcus and also the pre-occipital notch. Next, the subdivisions of occipital lobe. So the subdivision of occipital lobe consists of primary visual cortex and secondary visual cortex. Now let's look at the diagram. Look at the area of the occipital lobe colored in blue. So in this occipital lobe, you can see that there are a yellow lining called calcarine sulcus. So the primary visual cortex, which also known as V1 or Brodmann area, surrounds the calcarine sulcus on the occipital lobe's medial aspects. Or in other words, primary visual cortex is located at the lower bank and upper bank of the calcarine sulcus. Meanwhile, the secondary visual cortex, which also known as V2, V3, V4, V5, or Brodmann area 18 and also 19, surrounds the primary cortex and receives the information from it. So the information leaves the visual area is split up into dorsal and ventral streams. And fun fact about primary visual cortex, it is divided into 6 distinct layers. And there are 2 cells which is located in primary visual cortex, namely simple cells and also complex cells. The simple cells are highly concentrated in layer 4, meanwhile the complex cells can be found in layer 2, 3, and also 6.
Functions of primary visual cortex. The first stage of visual processing in the cortex is called V1. In primates, V1 creates a saliency map from visual inputs to guide the shift of attention or gaze. It does so by transforming visual inputs to neural firing rates from millions of neurons, such that the visual location signaled by the highest firing neuron is the most silenced location to attract gaze shift. V1's firing rates are received by the superior colliculus in the midbrain, which reads out the V1 activities to guide gaze shift. Functions of secondary visual cortex, or called V2. It receives strong feed-forward connections from V1, direct and via the pulvina, and sends strong connections to V3, V4, and V5. It also sends strong feedback connections to V1. In terms of anatomy, V2 is split into 4 quadrants, a dorsal and ventral representation in the left and the right hemisphere. Together, these 4 regions provide a complete map of the visual world. V2 neurons are also modulated by more complex properties, such as the orientation of illusory, contours, binocular disparity, and whether the stimulus is part of the figure or the graph. Functions of third visual complex, V3. Dorsal V3 is normally considered to be part of the dorsal stream, receiving inputs from V2 and from the primary visual area, and projecting to the posterior parietal cortex. It may be anatomically located in Brodmann area 19. Neurons in the area DM respond to coherent motion of large patterns covering extensive portions of the visual field. Functions of visual area V4. V4 is the third cortical area in the ventral stream, receiving strong feed-forward input from V2 and sending strong connection to the PIT. It also receives direct input from V1, especially for central space. Like V2, V4 is tuned for orientation, spatial frequency, and color. Unlike V2, V4 is tuned for object features of intermediate complexity, like simple geometric shapes, although no one has developed a full parametric description of the tuning space for V4. Functions of middle temporal visual area, or it's called MT, or V5. The V5 leads to deficits in perceiving motions and processing of complex stimuli. It contains many neurons selective for the motion of complex visual features, for example, lines, ends, and corners. Microstimulation of a neuron located in V5 affects the perceptions of motion. For example, if one finds a neuron with preference for upward motion in a monkey's V5 and stimulates it with electrode, then the monkey becomes more likely to report upward motion when presented with stimuli containing left and right as well as upward components. Functions of dorsomedial area, or called DM and V6. The dorsomedial area, DM, also known as V6, appears to respond to visual stimuli associated with self-motion and wide field stimulation. There are similarities between the visual area V5 and V6 of the common marmoset. Both areas receive direct connection from the primary visual cortex and both have a high melanin content. V6 is also sometimes referred to as the parieto-occipital area, or PO, although the correspondence is not exact.